Case 2, a bad mix. Jesse's dad consumes a few glasses of alcoholic beverages every night. Last night, his dad was complaining about the pain and decided to take a Tylenol with his usual drink. Does alcohol and Tylenol have an effect on catalase activity? Well, it is hypothesized that the catalase activity will be much slower in the Tylenol and ethanol solutions because catalase activity is most optimal at a neutral pH. According to that, the slowest reaction will occur when the solution combining both ethanol and Tylenol solution is reacted with the catalase because both Tylenol and ethanol are bases. The independent variables are the Tylenol solution and ethanol. For the procedure, 10 milliliters of hydrogen peroxide is added to the reaction chamber. Then a paper disc is soaked in catalase and stuck onto the wall of the chamber. To determine the catalase activity, the reaction chamber is put into a body of water with a tube leading to a graduated cylinder. Every 30 seconds, the displacement of water in the graduated cylinder is measured to determine the speed of the reaction. Moving on to the results. This table includes information such as the substance used, each trial, and the water being displaced every 30 seconds. To give you a visualization of the data, here are these graphs. These graphs show that the speed of the reaction is getting slower with each different test group. The fastest test group was with 10 milliliters of hydrogen peroxide added to the reaction chamber. The second fastest being hydrogen peroxide with 1 milliliter of ethanol, third being hydrogen peroxide with 1 milliliter of Tylenol solution, and the slowest reaction being hydrogen peroxide with 0.5 milliliters of ethanol and 0.5 milliliters of Tylenol solution. When hydrogen peroxide reacts with catalase, it speeds up the reaction, which decomposes hydrogen peroxide into water and oxygen. For the most part, the results collected from the experiment does match the hypothesis. Ethanol slows down the reaction because it is a non-competitive inhibitor. This means that it binds to and changes the shape of catalase, ultimately reducing its activity. Also, when high concentration ethanol is added to the reaction, it denatures catalase. The Tylenol solution slows the reaction because it contains acetaminophen, which inhibits catalase. In a functional human body, when there's a buildup of acetaminophen in the liver, the primary pathway to eliminate the byproducts are blocked and are forced to take an alternative pathway. This creates NAPQI, which is a toxic compound. The reason for the Tylenol solution and ethanol being the slowest reaction is because alcohol interferes with the way the liver processes acetaminophen. While performing the experiment, a problem that we came across is that the catalase will sometimes touch the hydrogen peroxide and start the reaction before it reaches the graduated cylinder. Something that would improve the design of the experiment is to have the hydrogen peroxide added to the catalase and have the reaction immediately transferred to the graduated cylinder. That's what's being shown in this diagram. This would help reduce the amount of oxygen gas escaping from the reaction chamber. The significance of this lab is to mimic a realistic reaction between the human liver and the mitochondria. The mitochondria releases hydrogen peroxide, which is toxic to the human body. So the liver decomposes it into water and oxygen, which is safer for your body to process. The lab was also executed to show how certain substances such as Tylenol and ethanol can slow down the catalyzation process of hydrogen peroxide in the liver. The results of this lab show the average person the negative effects that an excess consumption of alcohol and Tylenol can have on your liver. The next step of this lab would be to investigate other substances that could potentially speed up the catalyzation process in our livers to improve the decomposition of toxic substances to make them safer for the body to process.